Everyone. So I'm sorry again for all the technical problems that I had during my talk and that it delayed a little bit, but we are just 5 minutes late, and we have already ready from from Denver, Professor Allegra Aron waiting for us. So Professor Allegra, she works as a professor in University of Denver. I repeated, she's a chemist, isn't it, Allegra?
Yep.
And then has been working with metabolomics, and she works with a very special, for me, very curious technique about metal associations and chelating of metals and with metabolomics. And I'm really looking forward to learn about that because I don't know nothing about these topics. So Allegra we will talk, she will talk during 1 hour first, then we will take a 5 minutes break because she also needs a break after 1 hour talk. And then we will continue with the second part of her talk, okay? Allegra, welcome. The stage is yours, and please enjoy it.
Great. Well, thank you so much, Carlos, for that kind introduction. So yeah, as Carlos told you all, my name is Allegra, and I'm coming to you, I guess, virtually here from University of Denver, where I am an assistant professor in chemistry. And so I'm quite excited to be here today. As Carlos mentioned, in the second hour, I'll be telling you about some of the research that I did in my postdoc and also now in my own lab at University of Denver. But before we sort of dive a little deeper into the research, I will start in the first hour telling you a bit about mass spectrometry data acquisition. Okay, and so at the University of Denver, I typically teach a whole 10-week course on this topic, right? Ranging from chromatography to mass spectrometry and other sort of analytical techniques. And so let's see how this goes, fitting pretty much all of this into 1 hour. But hopefully it goes well. So I wanted to start with just the discussion of the learning objectives for today. As I said, we have a lot to cover. So I know some of your earlier talks were about experimental design, sampling, etc. So I hope now we're really ready to do our metabolomics mass spectrometry experiment. Okay, and so the learning objectives for this talk will be to understand first just what is metabolomics. I wish I could interact with you guys more to see who has actually done metabolomics and mass spectrometry, but I guess I'll just have to guess. I guess that's one of the issues with a virtual talk. Then the next learning objective will be to really understand different types of chromatography, different types of mass spectrometry, ranging from different ionization modes, different mass analyzers, etc., and how these can sort of have different strengths and weaknesses in metabolomics studies. And then finally, I'd like to think about which parameters are really essential to think about when setting up a non-targeted metabolomics experiment, both in the MS1 dimension and then in the MS2 or Tandem MS dimension. Okay, so how do we really best set up a mass spectrometer for the data that we want to acquire? Okay, but now let's just make sure we're all on the same page, right? So when we speak of mass spectrometry, we're really just describing the science of displaying the spectra of molecular masses of individual compounds and atoms as ions, okay? So a bit of a mouthful, but essentially we can think of a mass spectrometer as just being a very fancy and in some cases extremely expensive scale for molecules, right? So on the left here, we're looking at a molecule of domoic acid. When we run that through a mass spectrometer, we can see the high resolution mass, right? And then what we can do is we can annotate that with some molecular formula, right? Because that mass is just going to be a combination of different atoms, right? So carbons, hydrogens, nitrogens, oxygens, etc. And keep note of this molecule of domoic acid, as it will come back actually in my research talk. Okay, now mass spectrometry is really exciting because we can apply it to numerous fields, okay? And so just to motivate why this technology is really important, I wanted to give a few cases of the applications in which mass spectrometry can be very useful, right? So mass spectrometry can be used to, to find new natural products from plants, right? These are often bioactive molecules that we're interested in. Mass spectrometry can be used to basically understand how microbes communicate with one another in complex systems, right? This can be the human gut, or it can be the ocean environment, soil environment, et cetera. But we can use mass spectrometry to actually sort of piece out communication between microbes. We can also use mass spectrometry for proteomics, right? And so this is a course in metabolomics, but I didn't want to forget the fact that we can use mass spec to analyze proteins and also to analyze protein modifications and even how proteins and small molecules can interact. Mass spectrometry is also essential for quality control, right, in the food industry or in the pharmaceutical industry. And then also we can even start using mass spectrometry to explore what is out there in space, right? So there have actually been some mass spectrometry studies done in space. But now let's hone in on metabolomics. Okay, so what is metabolomics? Well, it is the large-scale study of small molecules or metabolites within cells, biofluids, tissues, organisms, or in a whole environment. Okay, and now unlike other omics measures, metabolomics is really unique because it directly reflects on the underlying state of the system, right? And because of that, those of us that do metabolomics, I think sometimes say it's one of the most powerful omics, right? Because it's exactly reporting on what is going on. Now, one thing though about metabolomics is it's essential to note that our experiment is strongly context dependent, okay? And that's dependent on your experiment and your experimental parameters. So what do I mean by that? Well, I mean that essentially the outcome of your experiment depends on your experimental setup, right? So do you choose an extraction with 80% methanol, 20% water? Do you choose an extraction with 50% methanol, 50% water? Do you add an internal standard? What sort of chromatography do you do? What type of mass analyzer do you use? Do you use electrospray ionization, or do you use MALDI, right? A laser desorption method. So these are many questions, right? And sort of the choices that you make actually will play an important role in determining what the experiment outcome will look like, right? Moreover, we have to decide, are we going to do a targeted experiment, right? Where we look for maybe one metabolite of interest. Say we're very interested in tryptophan. Maybe we just want to focus on finding that metabolite very carefully. Or do we want to look at everything in the system, right? And this would be more of a non-targeted experiment. Additionally, are we interested in quantifying the levels of our metabolites very precisely? Or are we simply interested in understanding what is there and what is not there, right? Now, this might be an important question if we have a wild type versus a genetic mutant organism. Okay, and so just one thing that I really want to make clear at the beginning of the talk, and I think you'll start to understand why this becomes important, is that we make a lot of choices in a metabolomics experiment, and it's really essential to think through those choices before we do our experiment, because those choices like extraction, mass analyzer, ionization, chromatography, et cetera, those choices will affect our data, right? And we want to be thinking about what our data will look like before we do the experiment, right? So we don't have to redo the experiment. Okay, so now I've told you a bit about metabolomics. Just to sum up what we've learned so far, metabolomics is the study of small biological molecules. Now, methods depend both on field and aim of study. Okay, and so the choice of instrument will impact the information content of the data. Experimental design before data acquisition is essential, right? And as I mentioned, there's no one size fits all method, right? We have to sort of develop our method with our scientific question in mind. Okay, now in metabolomics, we can use a variety of instrumental setups. So we can use a GCMS, we can use an LCMS, or we can use MALDI, mass spec imaging. Now, GCMS refers to gas chromatography. LCMS would make use of a high performance liquid chromatography system or an ultra high performance liquid chromatography system. And then MALDI is essentially a matrix assisted laser desorption ionization method that allows us to image various tissues, cells, etc., on a surface, right? So it's really sort of a different mode of mass spectrometry than LC or GC. And so we'll dive a little deeper mainly into these two LC and GC technologies now, and we'll really specifically focus on LCMS because this is what I do in my lab. I think this is what many of the speakers in this workshop do in their own lab. And also it's just very powerful for high throughput, non-targeted metabolomics applications. Okay, but in addition to these three modalities, there's also ion mobility mass spectrometry. And ion mobility, it actually looks at differentiating molecules by their collisional cross-section. And so this is sort of a really powerful emerging strategy for metabolomics. Okay, and so now let's think about what the data structure actually looks like in a mass spec experiment, right? So this might be a review for those of you who do a lot of mass spectrometry, but again, before we dive into more of the nitty-gritty, we all have to be on the same page with what the data will look like. Okay, so when we run our mass spectrometer, we are going to get a chromatogram that looks like this, right? Where essentially we have time on our x-axis and we have intensity on our y-axis. Okay, and so what we're looking at here is a number of features, right? And so a feature is simply a chromatographic peak with a unique retention time, peak shape, and mass to charge ratio. Okay, and so here we're essentially looking at increases in ion intensity with time, right? But then let's say we want to focus on what's going on here at maybe retention time 6. We can look at this time and then we can get a mass spectrum, right? And so this is an MS1, right? This is just looking at each of the intact masses that are eluting at this retention time. Okay, and we can assume this is a mass spectrometer coupled to liquid chromatography system, right? So here we have an MS1. And then let's say we're interested in one of these abundant peaks in the MS1. Well, then we can look at sort of the next dimension, and that is the MS2 or the tandem MS. And we'll talk about that in just a few moments here. But this is essentially going to be the fragmentation spectrum associated with this intact molecule. Okay, but yeah, we'll go into some more details on that at the end of the talk. Okay, so now before diving into the mass spectrometry part, let's think a little bit more about the various techniques that we use for separation, right? So mass spectrometry in itself is a separation technique, right? Where we separate our ions. And remember, in mass spectrometry, we're only looking at ions, right? We're looking at charged ions, right? So mass spectrometry, we can separate those ions by mass to charge ratio or M/Z, but to sort of make it so that our mass spectra are less hectic, more simple, we often couple our mass spectrometry system to a separation technique that occurs beforehand, right? And so that could be a mass spectrometry or a mass spectrometer coupled to gas chromatography system, to a liquid chromatography system, to an ion mobility spectrometry system, right? And so gas chromatography then will do separations based on boiling point, whereas liquid chromatography separations will be relying on hydrophobicity or polarity, right? Interactions between our analytes and a solid phase that we'll talk about again in a few slides here. And then this separation will be manifested as differences in retention time or elution time. Now, ion mobility separates based on a rotational shape or size in the gas phase. This is that collisional cross-section that I referred to a slide prior. And here, our separation depends on or is manifested as an ion mobility or CCS, collisional cross-section. Okay, and so we have all of these different separation modes. The question then is, why do we use one versus the other? Well, sometimes it's just what we have available to us, but let's say we have everything available to us. Now, gas chromatography, liquid chromatography, and ion mobility really have different strengths and different weaknesses. So gas chromatography is really good for looking at compounds that are volatile, right? Because as I told you, gas chromatography separation is based upon boiling point. Okay, so it's looking at these volatile compounds. Typically, volatiles can be small and nonpolar. When I think of GC and compounds that are really great for GC, I often think of short chain fatty acids, and maybe this is because I'm coming from the microbiome field, sort of, where people are really interested in these short chain fatty acids. But you can think short chain fatty acids are small, they're nonpolar, they're volatile, right? Now, let's say we want to look at larger or more polar molecules. We can actually do a chemical derivatization of these molecules that will make it so that they can actually be volatilized more easily, right? But this adds an additional step of derivatization. All right, now how about HPLC, high performance liquid chromatography, or ultra high performance liquid chromatography? And now what is the difference between HPLC and UPLC? Well, the ultra high performance runs at a higher pressure. Okay, and so this is really good for sort of nonpolar molecules. It's really good for polar molecules. Also, maybe molecules that are slightly bigger or less volatile than those molecules that we often look at in GC. It's also really good for macromolecules, okay, or proteins, for example. And so something that's nice about HPLC, also GC, but there are a variety of different modes for separation in HPLC, right? So we can do reverse phase chromatography, which is using a non-polar resin. We can also do normal phase or Hillick separations, which use polar resins. And then in a slide or two here, I'll tell you about some other strategies that we can use for liquid chromatography, such as size exclusion, affinity chromatography, etc. And then we have MALDI, right, which can be really useful for samples that are quite different from the samples that we run typically in HPLC, right? So for HPLC, we'll typically do an extraction. I suppose, or I imagine you talked about this in some presentations prior, right? Where we have our sample, we add some mixture of water and methanol often, then we do some sort of homogenization, right? We get out our extract. But in MALDI, we can actually do our imaging experiments on tissues directly or in some 96 well plate, for example, in that format, or we can use solid samples or spots. And so we can actually sort of directly do that analysis without the need for as much of the extraction as in HPLC. Okay, and so now I think this will probably be a review, but let's just think a bit about what is chromatography, right? So again, you've probably seen these experiments. Maybe you saw these experiments back in your high school or college days, right? Because this is actually an experiment that I run with college students, right? Where we take just some markers, right? And we can see the dyes separate out, right? So this is an example of either thin layer chromatography or paper chromatography, right? Where we essentially get our dyes moving at different speeds based on their interaction with the paper. Now, super basic example, right? But we can sort of go up in complexity, but the principles stays the same, right? Where here we're actually looking at sort of a solid or liquid versus gas extraction, right? And this would be what we would use in GC, where in gas chromatography, we're actually using an inert carrier gas as our mobile phase. And then we have some liquid or solid stationary phase, okay? Now in liquid chromatography, as I described, and we'll be showing you on the next slide, we have a solid stationary phase, and then we have a liquid mobile phase, right? And the separation will be based on interactions between those phases. Okay, and so what are these interactions? Well, we can look at polar interactions or non-polar interactions. We can look at ion binding or exchange interactions. We can look at differences in boiling point. We can do separations based on size, right, where we have size exclusion chromatography, or even affinity chromatography, where we may have some metal or some ligand on the column, and that will actually be sort of mediating the separation. Okay, but now I'd like to talk a bit about reverse phase chromatography, because I think for exometabolomics studies, you'll likely be doing a lot of reverse phase chromatography. And so here we essentially have a stationary phase. This is going to be our nonpolar phase. So I think many of you have probably heard of different stationary phases, including C18. So that's what it looks like, right? We have our silica here, and then we have that sort of appended to a long C18 chain, right? We also have a variety of other stationary phases for reverse phase, including C8, right, which has a 8-carbon chain, phenyl, which has this phenyl ring here, and then we have a variety of sort of more fancy and more efficient C18 types of resin. So here we're seeing a C18 shield, and there are a variety of column vendors that I just kind of wanted to bring your attention to, because each vendor sort of has their own sort of spin on different sort of resins. But the basic principle is the same, right? We have some stationary phase, and this is nonpolar, and then we flow our polar mobile phase over the stationary phase, right? And then molecules either interact more or less with the stationary phase, and these interactions determine the retention time at which we see the molecule, right? And so chromatography will be one area that you'll likely want to spend some time optimizing for your exometabolomics experiments, right? And so let's think a bit about what we'll actually be trying to optimize when we do our chromatography, right? So essentially, we want our peaks to be as sharp or resolved as possible, right? And so what does this mean? We'll talk about that in a second, but we can think about an experiment, right? So this is just a diagram coming from a basic analytical chemistry textbook, right? But when we start our experiment, everything is aligned as a very sharp peak, right? But then we get peak broadening as it goes through the column, right? And so here we still have a peak that's relatively sharp, but now as we continue going through the column, this peak broadens, right? And so what is sort of a nice peak shape versus what is too broad? Well, here we have sort of a diagram showing a nice Gaussian shaped peak, right? And so we can start looking at sort of different parameters or different feature or different points on this peak to sort of think about what is the ideal peak shape, right? And so here we have our T0, right? This is our time of injection. Here we have our retention time. And then here we have our width at half of the height. And then here we have our peak width, okay? And so now we can start describing different characteristics of the peak, right? So we can think about a retention factor, K, right, which is essentially the difference in our peak retention time minus the starting injection time divided by the injection time. We can also think about a selectivity factor, right, where this would essentially describe how selective our column is for differentiating between different analytes. And then we can start describing the resolution of the peak, okay? And now we don't have to spend too much time thinking about these factors, but these are going to be important factors and parameters when we're thinking about how to best set up our chromatography, okay? And so here, this was supposed to be an example of a nice chromatogram, but somehow some of the baseline has gotten cut off. But what do we mean when we say a nice chromatogram? Well, you can see here that we see sharp peaks, okay? And so these peaks are not broad, right? Rather, they are sharp, they are resolved from each other, right? And when I say resolved, I'm talking about resolution, as I described on the last slide, right? We can see these two peaks are distinct from one another. And if you could see the baseline, you would see that the baseline is flat, right? And there's a high signal to noise, right? We can't see the baseline because, well, something happened with this image, but also it's very low. Okay, and now that we sort of have an understanding of different separation techniques, right? Whether we're using GC, LC, ion mobility, etc., we can now start to think about how those separation techniques will be used in conjunction with mass spectrometry. Okay, and so here it's useful to start thinking about a mass spectrometer as sort of a very simplified system, right? Where we have our inlet, we have an ion source, right? And in this ion source, we have to somehow make ions, right, from molecules that were floating around in solution, not as ions. Okay, now from this ion source, then once we have ions, then we have our mass analyzer. Okay, this allows us to separate out our masses. And then from there, we can actually detect what masses, or rather what are the M over Z's that are actually there. Now, underlying our mass spectrometry system, we have a lot of electronics, and we also have a very good vacuum system. Okay, so it's important to note that all of this occurs under vacuum, right? So having a powerful vacuum really underlies our ability to do mass spectrometry. All right, and then what do we get out from our mass, rather our computer that's reporting on what it sees? Well, we get out a retention time, we get out an M over Z or a mass to charge, we get out an intensity, and if we have an ion mobility dimension, we can get out that collisional cross section. Okay, and so this is kind of like conceptual. Now let's start looking at really what are the different ion sources we can use, what are the different mass analyzers, what are the different detectors, and why might we want to use one versus the other. But before we dive into that, let's just think a little bit about some fundamentals, right? So when we actually do a mass spec experiment, we're often collecting in profile mode, and that's what you see here. But I think when many of you have looked at a mass spectrum, you're more used to seeing this, right? And this is because we typically look at mass spectra in centroid mode. Okay, and so the profile mode data detected by the mass spectrometer actually consists of distributed signals, as you can see here, across M over Z values at each point where an ion is detected. Now, centroid data is the raw data that's processed to retain only the local maximum in each range in which an ion is detected, right? And so that's what we see here, right? We're looking at the local maximum. Okay, and now additionally, we also need to take into account the fact that in our mass spec experiment, we are looking at a variety of isotopes, right? So recall, we're looking at C12 isotopes, C13 isotopes, and this can become even more complex for metals. That's part of what I'll be describing in my next talk in a few minutes here. So yeah, stay tuned for that. We can also have complex isotopic patterns when we have halogens and many other elements as well in our molecular formulas. But now, in addition to isotopes, we also can start to see a variety of adducts and in-source fragments that are associated with sort of our feature of interest. So let's say we're looking for this molecule and we're expecting to see the M plus H, but then we see in our MS1 a variety of other peaks. So that could be because we have water losses, we have adducts, right? We have, and we actually have a variety of adducts, right? And so when you start looking at all of the features in your data set, it's really important to think about the fact that each feature is not necessarily a unique compound, or is not necessarily, a unique compound, right? We could have maybe like 5 or even more features associated with one molecule, right? So with like tryptophan, for example, we could be seeing the proton adduct, the ammonium adduct, the sodium adduct, etc. And so it's important to try to link these adducts together. And I think through this course, you'll start learning about methods that facilitate this. Additionally, in mass spectrometry, we can start seeing multiply charged species, right? I'm not going to spend too much time describing this, but we can essentially take this and do a charge state deconvolution that allows us to look at the neutral mass of each of these peaks. This is most commonly observed when we're doing proteomics. In metabolomics, it's usually common to see singly charged, doubly charged, and maybe a +3, but typically we're looking at 1 or 2 charges. This is very important for proteomics, though. All right, so now let's start thinking about sort of the differences in mass spec instrumentation, right? And so there are a variety of instruments, which I'm sure many of you are aware of. We have triple quad instruments, we have ion traps, we have QTOFs, or quadrupole time of flights, we have orbitraps, and then we have Fourier transform ICRs, right? And now we're going to come back to this slide, but just one thing that I want to get you thinking about is the fact that, again, there's no perfect instrument, right? There's no instrument that's just the best, right? It's sort of dependent on your application, right? We can start to see that each of these instruments have very different capabilities in terms of resolution, in terms of accuracy, in terms of linear range, in terms of speed, and really in terms of cost, right? And so in the next few slides, we'll think about what is resolution, not in the context of chromatography now, but rather in the context of mass spectrometry. Why might we need linear range, etc. And then hopefully that can start to guide your thinking about which mass spectrometer and which mass analyzer might really be perfect for your application, right? And so this is just kind of a funny way of saying we can sort of think of each mass spec as like a Pokemon card, right? Because each mass spec has some strengths, but also some weaknesses, right? And it's sort of like which one is the best for your application, right? That depends on resolution and accuracy, sensitivity, or linear detection ability, scan speed, costs, etc. Okay, and so we talked about resolution in the context of chromatography. Here in the context of mass spectrometry, we can sort of look at how differences in resolution really affect what we're looking at, affect our peak, right? And so we can look at with a very low resolution, or this is not that low of a resolution, but with a relatively low resolution compared to these higher resolutions, our peak is broad, right? And how are we defining resolution? Well, we're defining it using this formula here, where we're essentially looking at the mass divided by the difference in mass at the feature width half max, okay? And so what you can see then, as makes sense using this formula, when we have a very high resolution, our peak width is more narrow. That peak is what we say, we call it a resolved peak, right? We are very certain that that mass is here at 195.0877 with a very tight mass tolerance. Okay. And now we can start to compare resolution and mass accuracy across various instruments. Okay, so with a low resolution, with a low resolution instrument like a triple quadrupole, our mass accuracy will be sort of, we can really only report it to this position, right? But as we go to increasingly more, or as we go to instruments that have increasingly better resolution, we can start reporting more decimal places with more confidence, right? So looking at an Orbitrap instrument, for example, we typically are looking at mass accuracy of about 5 ppm or less, okay? And when we say this, we're really thinking about the calculated mass as compared to the measured mass, okay? And like specifically, what is that difference? Okay, and then here is just sort of another way of looking at differences in resolution, right? One of the things we note is at a very high resolution, our ppm error is lower, and we can differentiate between peaks that are very close together, right? So one of the examples that we often use is when we're looking at a low res instrument, sometimes we can't distinguish between compounds that have different molecular formulas, but the same, but very similar or the same within unit resolution weights. Using a high resolution instrument, we can. And that's why we want to use very high resolution instruments when we're trying to annotate the molecular formulas of unknown compounds. Okay, and so now that we have sort of those basics down, like what is mass resolution, et cetera, then we can start again breaking down those components of the mass spectrometer, right? And so the first component is sort of the ion source, okay? And so here we can start to see different ways of ionizing our compound, right? And so we have ESI, which is electrospray ionization. We have APCI, atmospheric pressure chemical ionization. And then we have GCMS. This should actually say GCEI-MS. This would be electron ionization, okay? And we'll talk about what are the differences in these different ionization modes, right? But one thing to note is that ESI, electrospray ionization, is useful across a very wide range of compound sizes and polarities. And that's one of the reasons that ESI, or electrospray ionization, has gained such popularity since its advent back some years ago. Okay. And so this is that diagram that I showed previously, looking at GC compared to LC. Now in GC, we typically couple gas chromatography to electron ionization. And this is what we call in chemistry a hard ionization mode. Okay, and what does that mean? Well, essentially it means that we're giving a lot of energy in our ionization to our molecule, right? And this actually kind of busts our molecule apart, right? And so we see a number of fragments and we see the M plus dot ion. We don't see the M plus H as we see in the case of electrospray ionization, right? And so one positive about using electron ionization is that this is standardized. We're always doing our electron impact at 70 electron volts, right? And so we have a very standardized way of looking at compounds. A drawback is that we don't have our intact precursor mass usually. And so this can make it hard to know what sort of the intact compound was. And so to get around this, we can do chemical ionization. We won't go too much into the details of this, but chemical ionization allows us to see that M plus H plus or M minus H minus precursor. Now, electrospray ionization, in contrast, is what we call a soft ionization strategy. This is very commonly, probably most commonly, used with HPLC or UHPLC. And it allows us to see an M plus H plus or an M minus H minus, in addition to those other adducts that I was discussing. Now, MALDI is also a soft mode of ionization. And when I say soft, I mean that we're not busting our molecule apart, right? We're seeing our intact M plus H. And then when we are sort of breaking our molecule apart, that's actually inside the instrument, right? It's not at the source. That's in a separate part of the instrument that I'll be talking about in a second here. Okay, so now let's have a look back here. So we very briefly talked about how we form ions. Now we can think about our mass analyzer, right? And so when we think about a mass analyzer and a mass detector, that's where we start thinking about these different instruments, right? So one type of mass analyzer is the Quadrupole, right? And this Quadrupole is very, very useful. It's used in this triple Quadrupole instrument. It's also used here in our QTOF, right? The Q in this time of flight stands for Quadrupole time of flight. It's also used in a number of orbitraps made by Thermo, right? That would be a Q orbit trap. And so let's, in a minute here, talk about what is this Quadrupole? Why is it useful? So here you can see what a Quadrupole looks like. They actually are very small, right? Look pretty cool. But yeah, interestingly, many of these parts of the mass spec are actually smaller than you might think, of course, if you look at the very big mass spec. But here's the Quadrupole. You can see it scaled next to a watch. Okay, and so how does a Quadrupole work? Well, let us take a look at it. Many of you have probably heard of dipoles, right? So a Quadrupole is essentially two dipoles, right? And so we have these charged rods sort of lying in the formation that you can see here, right? And so we have alternating charges. We have a positive charge, negative charge, positive charge, negative charge. And so why do we do this? Well, that creates an electric field here, and using and modulating that electric field can actually allow us to modulate how the ions that we've introduced into our Quadrupole can actually move. Okay, so what happens? Well, first, our ion enters the Quadrupole system, right? It's positively charged, as we know, like charges attract, like charges repel, opposite charges attract, right? And so electrical repulsion and attraction, respectively, between the Quadrupole ion and rods occurs, right? That moves the ion in this direction. But then what do? Well, we can actually swap the RF voltage. Right now, this changes the polarity and the electrical repulsion between the quadrupole rods and the ions, and that actually changes the way that the ion is moving, right? And so now the ion movement has changed, and again, the ion is moving in the direction of the nearest quadrupole rod with the opposite charge. We can flip this again, and now that changes the flow of the ion again, right? And so what we can do essentially is we can sort of direct our ion of interest specifically through that quadrupole, while all of our other ions, the ions we are not interested in, can sort of go off and will not be allowed to pass through, right? And so depending on how we modulate our RF voltages, that depends, or that allows us to let through certain things. Sometimes we just want to let through all our ions, etc. But the really nice thing that we can do with a triple quadrupole instrument, and this is why we can use triple quads really well for targeted experiments, is we can actually do this multi-reaction monitoring, or MRM scheme. And so how that works is we essentially take our we essentially take our first quadrupole, we use that as a mass filter to allow a fixed MZ value through. Here in our second quadrupole now, we've actually set that up to actually fragment that molecule, and we'll talk about fragmentation in a minute here. And then in our last quadrupole, our third quadrupole, we can essentially let through another fixed value. And here we're detecting that fixed value, and that can allow us to say, okay, we see our molecule of interest, right? Now, this is also known as selection reaction monitoring, right? And so lists of transitions can be cycled through. And so this allows for very high sensitivity, okay? And so even though triple quadrupoles do not have the mass accuracy and resolution of Orbitraps or QTOFs, they have very high sensitivity for quantification. And that's why these instruments are very ideal for quantification. Okay, now moving right along, because we still have lots to cover. If we're interested in doing a non-targeted experiment where we want to be looking at many analytes and we want to be looking at them with a high resolution, we can make use of a time of flight, or a TOF analyzer, or an Orbitrap analyzer. Okay, and so this TOF is actually coupled often to a Q, a quadrupole, before the TOF. Now, what does the inside of a TOF look like? Well, here we have our source, right? This would be an ESI source. Now you all know about ESI, a soft ionization mode. Then we have a quadrupole, and then we have our sort of flight tube here with a reflector that allows us to actually reflect the ions. And this makes our flight tube even longer, right? It's twice the length, right? And so that's why people, or that's why instruments often make use of these reflectors. Okay, now how does a TOF work? Well, this is sort of the canonical way of mass detection, right? And so what we can do is we can start with a packet of ions. We give these ions the same kinetic energy. And then what we can see is the smaller ions separate out first, and the bigger ions kind of are left behind, right? And this allows us now to separate those ions based on mass to charge ratio, right? Essentially, when they hit the detector, we can look at the relative abundance of each of those ions as well. Now, another mode for detection is using either an Orbitrap or an FTICR. Now, an Orbitrap contains this very small trap. An FTR contains a very large and powerful magnet, but essentially what we start seeing is ion oscillations, right? And so sadly, I don't have an animation, but when we introduce packets of ions into this trap, we start to see that they actually oscillate around and back and forth, right? And that oscillation frequency is determined by the mass to charge ratio of the ions, right? And so that's what we're looking at here. We can do a Fourier transform, right, which is a common strategy used in mass spectrometry and in nuclear magnetic resonance, NMR, right? And from there, we can get out our mass spectrum. Okay, and so this is how an MS1 scan would look in the Orbitrap. We'd have our chromatography, ESI, a quadrupole, and then here you can start to see that oscillation, how that generates this spectrum, and then how we do a Fourier transform to actually get out our different masses. Now, what are some things to note about these trapping systems? Well, they're very high resolution, right? However, we want to be careful not to introduce too many ions into our system. This was a funny video that's not quite working. Let me see. Here, okay, here we go. So we can sort of just go through the video quickly, and what you can see is here we have a bunch of ions. Now, what happens when you try to push them all through? Well, they each affect one another, right? And so this is just a funny example of how space charge effects can occur, right? If we pack in as many ions as we can into our trap, well, the actual charges of the ions start affecting one another. And this actually starts affecting our mass accuracy and resolution. And so we want to be careful to not be sort of packing our trap too full, okay? And so in order to do that, we have this security guard there. We can call him automated gain control, right? And he actually sort of uses the C-trap, which is essentially a holding point before the Orbitrap to basically stop when before there are too many ions in the trap. Okay, so now that we've kind of gone through the different ionization modes, analyzers, and detectors, all sort of at whirlwind speed, let's think a bit about this MS2 dimension. And so what is this MS2 that I'm speaking of? Well, tandem mass spectrometry is essentially when we choose one molecule of interest and we actually apply energy to that molecule inside the mass spectrometer, right? Breaking it apart into its fragments, right? And so now we've broken that molecule of interest into its fragments, into its pieces, essentially like if we have a puzzle, we throw it on the floor, we now have a variety of puzzle pieces. And from there, we can get that fragmentation spectrum, which is called an MS-MS spectrum, okay? Here, we're looking again at that favorite molecule, domoic acid, and we can see where we might be fragmenting it. Okay, the cool thing is we can do that first fragmentation, which we call MS2, but then using some trapping instruments, we can actually do an MSN. That allows us to basically, we have our MS1, we can do an MS2, and then if we have a fragment that we are like, oh, I want to know more about what that fragment is, we can actually do an MS3, even an MS4. Now, this is not possible on all instruments, okay? So like the instrument I have in my lab, which is a Q-exactive HF Orbitrap, you can only do MS1 and MS2, but some ion traps allow for further fragmentation. Okay, and so before we kind of dive into how we might want to actually set our parameters for our MS-MS acquisition, let's just think about why we actually would want to do MS-MS. Well, we've talked a lot about MS1, right? And how if we have a high resolution mass, we can come up with a precise molecular formula. But let's say we want to start thinking about how, well, what is the connectivity of the molecule, right? We can have a molecular formula, but we don't know molecular structure, right? So MS2 allows us to start thinking about what is the connectivity of the molecule, what might actually that compound be, okay? But from an MS2, we can have variety of confidence that our sort of annotation or saying this spectrum is this is correct, right? And so a level 1 annotation means that we have actually matched our MS2 with an authentic standard, right? So we have that authentic standard, but we also have another piece of evidence, and this could be a matched retention time, this could be a matched NMR spectrum, etc. Then we have a level 2, which means we have just an MS-MS match to that compound. Then we have sort of medium level of confidence. This could be an annotated formula match or a class match, right? So we're not saying we have tryptophan, rather we're saying we have that molecular formula and we think it's an amino acid. And then we have low confidence, where we simply just have our mass and our spectrum, but we don't know what it corresponds to. So this is really useful, and I would really encourage reading more about this. But how do we choose which ions to fragment? Okay, so we're running a little tight on time, so I might sort of skip over a few slides, right? But we've done our experiment, right? We see many ions. How do we know which ones to fragment? Well, typically in my lab, and I think the labs of some of the other presenters, we perform data-dependent acquisition. And essentially what this means is we use the data to tell us which MS2s we want to acquire, right? So we take an MS1 scan, and then what we do is we choose some top number, so top 5, for example, most abundant MS1 masses, and then we further fragment those, as you can see here. And this is what it looks like on the instrument that we have in my lab, the Q Exactive HF, okay? So what you can see is we do one MS1 scan. You'll see that in a minute here. And then we go and do a variety of MS2 scans after that, okay? Now there are other ways of doing this, right? So this is the DDA approach, the data-dependent approach, where we select some top 5, fragment those, but we can also do data independent, where we're not using our data. We're simply saying that we want to fragment across all isolation windows. So like this blue chunk, this red chunk, this green chunk, etc. That allows us to get a number of MS2 scans, right? But we can also have issues with chimeric spectra, etc. Okay, and so now we might look at 2 different methods and say, well, wow, method A, we don't see that many MS-MS triggered, but method B, we see a lot. So which one is better? We might think, gosh, method B looks a lot better. But then we realize, well, what are all of these MS-MS spectra that are triggered? Well, maybe we're just triggering a lot of the same compound. And unfortunately, maybe we have plasticizers or other contaminants that are really abundant in this sample. So then in this case, this method B does not look so good, even though we see a lot of MS-MS spectra, right? So maybe we could put these on an exclusion list, which means that we would essentially tell the instrument, okay, let's not collect MS-MS of these contaminants and plasticizers, okay? Now I'm gonna skip through these slides because we are running a little short on time, but I did want to finish just by highlighting a paper that came out from Daniel's group, and I played a small role in this too. So, but the reason I want to talk about it is because I think it's a super, super useful paper when you're getting started thinking about, gosh, like there's so many parameters on an instrument, like here, a Q Exactive, top N, isolation with apex trigger, monoisotopic selection, et cetera. What do these all mean? Where do I start, right? So this paper does a really nice job of laying out what do each of these mean, and what is actually a good starting point, okay? So I wanted to go through just a few of the parameters that were tested in this paper. So the authors of this paper did a really nice job of going through systematically and looking at how changing these settings with a variety of parameters actually affects the data. Okay, and so we've talked about resolution, right? So one of the key parameters to set is the mass resolving power, right? And this influences how well M over Z signals are resolved, right? And I'm throwing tons of information at you, but this sort of looks similar to the picture that I showed a few slides back where we were looking at different resolutions, right? So this purple, we have a very high resolving power, right? And so as you can see, when we increase our resolving power, we're actually seeing fewer nodes, and we're seeing the annotation rate also changes, right? And so mass resolving power clearly has a big effect on our molecular network. Okay, the next thing to think about is that automatic gain control. And if you remember what AGC is, well, that was the big bouncer guy that I showed you, right? And so AGC represents the number of ions filling the C trap before they're transferred into the orbit trap, right? So we want to get as many ions as we can, but also we don't want to be seeing space charge effects, right? And so we have to set our AGC in a controlled manner, okay? And so we can see that AGC also is going to be affecting the outcome of our data. Okay, now finally, I would like to mention this idea of microscans, right? And so microscans are essentially the number of internal scans that are merged into written scans, right? And I also want to think about the fact that we have to set a duty cycle time, right? And so here, for example, we see varying duty cycle times, right? So if we do an MS1 and collect the top 3 MS2s, our duty cycle time might be shorter than if we do an MS1 and collect the top 15, right? And so again, balancing what we want in our experiment. If we want to collect the top 15 MS2s, we can see that our we're not scanning as frequently, right? So our peak shape doesn't look great, but at the same time, we are getting all of those MS2s, right? So if that's kind of what we want in our experiment, then, it's a trade-off. So it's really important to know how these parameters are going to be affecting sort of what we want in each of our experiments. Okay, finally, we can look at dynamic exclusion, right, which actually excludes the precursor MZ after we've collected for some specified time frame after the first MSMS experiment. And you can see how different settings for dynamic exclusion really affect differences in the amount and specifically which MS2s are required. Okay, now I think this is the final parameter that I'll go through, and I know I've gone through these really fast. I really would suggest looking at the paper, but we can set an isolation width, which defines the MZ range around the precursor ion for MS fragmentation, right? And this will sort of unsurprisingly affect the precursor purity, right? So this would be the intensity of a selected precursor divided by the summed intensity of the isolation windows. And so I know I really quickly just breezed over those parameters. There's a lot there, but I did want to point you to a variety of papers that actually go through in good detail the parameters for a variety of instruments, right? So I focused on this paper because, well, me and various other people that are speaking at this workshop were involved in this paper. And also it's on the Q Exactive, which is the instrument here that I have in my lab. But if you have a different instrument in your lab, I would really recommend you to look at these other papers, right? Where we can understand what parameters are ideal for doing exometabolomics on a QTOF or on various other instruments. Okay, and so I think I've gone just a bit over time. I am really happy for this invitation. And I just wanted to thank all of the other speakers, the organizers, specifically Carlos for the invite. And I also really want to thank Daniel Petrus, who's going to be speaking, I think on Thursday, I believe, or tomorrow. And also Corinna, they gave me a lot of slides that I modified. But the slides were a really good starting point because this is really a lot of information to condense into 1 hour. So I hope you've enjoyed that. I don't know if we want to take a few questions or if we want to take a break. I'll leave that up to you, Carlos. And then we can do my research talk in a minute here.
Yeah. Thank you very much, Allegra, for your nice talk. So, do you listen to me, Allegra?
Yeah, I can hear you.
Yeah, okay. So, thank you very much. I think we have time.
Thanks.
Very quick for a couple of questions. If you have some questions, can you please use this microphone?
So I have a specific question about the data acquisition for non-targeted metabolomics, right? Because most of the times you see papers on one technique, right? Using LCMS or GCMS only, NMR, etc. But you don't see very often the combination of multiple techniques, and I wonder, because every technique has particularities, as you explained, how much are we missing by not, using all possible techniques that we have?
Yeah.
And also considering redundancy, right? Because if you use LCMS, for example, and you acquire negative, positive, but also GCMS, then you will be gaining, but maybe not gaining too much, and you have a lot of redundancy in your data, right?
Yeah, so I think that's a great point, right? Because I think the ideal would be to do like LCMS, positive mode, negative mode, reverse phase, Hillock, to do GCMS as well, maybe even to do NMR metabolomics, right? But I just think the practicality of it is that most people would not do all of the different analysis modes, right? And so we typically try to do reverse phase, positive and negative mode, and we're starting to try to do Hillock as well, because I do think we focus on reverse phase often because we're interested in microbial metabolites, secondary secondary metabolites, right? Natural products. But by focusing on reverse phase, we're missing a lot of the more polar metabolites that we might find in Hillock, right? And so I think it is important to do sort of a variety of the different strategies if we want to do a fully non-targeted experiment, right? But for example, if we're interested in like, as I'll talk about in my next talk, metal binding molecules specifically, and we look at the genome, right, and we think we know the structure of the molecule, that might be when we can take not really a semi-targeted approach, but we sort of think we know what the molecule will look like, and so we can say, okay, reverse phase seems like it'll be a good strategy. This molecule might have groups that ionize in positive or negative mode, etc. So I'm not sure if that fully answered your question, but I think it's a hard question, but a very good point to make.
Thanks a lot.
Yeah. Oh yeah, you can write me in the chat as well.
Do you listen to me now?
Oh yeah.
Allegra, what about 5 minutes? Are you okay with that?
Sure. Yeah, that sounds
5
good.
minutes break, and then we can go to the toilet, and then we continue with Allegra.
Perfect.
Thank you very much.
Awesome. Okay, thank you.
Thank you.